Okay, so earlier today when I was giving this Zoom lecture, I actually forgot to record the first third of it. So this recording now is actually one that I've done again. So for those of you who actually tuned in live, it's the same content, but that's why some things might seem a little different. So what we're talking about today has to do with this interesting sort of thought-provoking concept of relating the size of an organism and its lifespan to the threat of cancer, and how organism size and its lifespan are really factors that Mother Nature has considered when equipping different organisms for how best to deal with the threat of cancer. So just to review some key concepts that we'd been talking about before spring break, we were talking about how telomeres are these ends, these sort of dispensable free end pages of our chromosomes. So every chromosome is one long strand of DNA, and that means at the ends, if you don't have any protection for them, you'd have these unprotected free ends of DNA, which can cause all kinds of problems. So telomeres are these repeating units of TTAGGG that repeat for, let's say, 10 kilobases worth at the end of each of our chromosomes. And importantly, they protect our genome from chromosomal damage, so that the ends of our chromosomes aren't just sort of destroyed and eaten away like the frayed ends of shoelaces. They also prevent chromosomes from being damaged by preventing them from sticking together end to end. So you don't have two different chromosomes suddenly fusing, especially during mitosis when the sister chromatids are really close together and held close together on purpose. And importantly, having these extra lengths of DNA at the end of the chromosomes are physical platforms where DNA replication is allowed to happen. Remember that the lagging strand of a copied DNA molecule that's being made depends upon short RNA primers to be able to have something to sit on so that you can then start making an Okazaki fragment, right? So the telomeres then provide this extra space where these primers can dock, so you can make sure to copy everything in between. However, that means with every cell division, you're going to have the very ends of the telomeres being left out from being able to be copied. So with every cell division, the telomeres get shorter and shorter and shorter. And evolutionarily, this is useful because we can use this as sort of keeping a tally or keeping track of how many cell divisions something like a fully differentiated cell has gone through. Because once those telomeres get too short, then we say that the cell faces crisis. So normally when a differentiated cell encounters crisis, that's a good sign that it's probably done proliferating. It shouldn't worry about dividing anymore, and then it might actually just go ahead and senesce, right? Most likely the differentiated cell at that point is just simply physically lost from our body. So we don't worry about the fact that along the way it might have built up some mutations. As far as cancer is concerned, then, we learned that overcoming this crisis and overcoming senescence are required for the cancer cell to achieve immortality and keep dividing well beyond what it should as a normal, healthy, differentiated cell. So here's a thought question then. Do telomeres behave the same in other animals? So for example, this mouse here, which is a mammal just like us, you would think, well, if telomeres play such an important role in our cells, they should also play an important role in the cells of a mouse as well, right? And so we'll pretend today that this little mouse is joining us in class, and he wants to learn about telomeres, among other things that are important that mice want to know about. So let's think about this. When you look at a mouse, you can think about things like the size of the mouse, its body size, and its lifespan. So the body size of a mouse, a typical mouse is maybe like 40 grams. In contrast to a typical human being, we might be 70 kilograms, right? So something like 140, 150 pounds. That means there's a difference of about 2,000-fold in size between a human and a mouse. Why is that important? Because you can use that 2,000-fold as roughly the size difference of how many cells make up the body. And that means how many cells might potentially turn into cancer that you have to worry about. Another thing to think about is the lifespan. So a typical mouse, maybe living in the wild, would be expected to live maybe 1 to 3 years, for example, before it's eaten up or it just goes ahead and dies, right? Compare that to what we can expect these days for a typical human being to live, maybe 70, 90 years or even greater. So we're talking about a 30-fold difference in the lifespan. Why is that important? Because remember, we learned that as a cell is transforming and heading towards cancer, it's building up random mutations over time. And it takes time to build up the right combination of mutations by chance for it to really become full-blown cancer, right? So the longer the organism is alive and around, the greater time there is for these mutations to build up. So we say body size is, you can think about it in terms of number of cells in the body that can become cancer, and lifespan, the time for mutations to actually pile up. And so this is actually very thought-provoking then, because if you go ahead and you look at the telomere size of a typical mouse, mice telomeres are about 40 kb in size on the ends of their chromosomes, compared to human telomeres, which are about 10 kb. So there's about a four-fold bigger telomeres in the chromosomes of mice compared to us. So this might seem kind of curious, right? Because it almost makes it look like that the clock for differentiated cells in mice to keep dividing and dividing and dividing and dividing is much more lenient than the clock inside of a human. Almost to say that in a mouse, there isn't as much concern that a differentiated cell might start building up and accruing mutations over time. Why is that? Again, it has to do with two things. A mouse has far fewer cells in its body to start with, so fewer opportunities for a cell to start going bad. And importantly, the mouse's lifespan, right, is only about 1/30th of that of a person. So therefore, even if you have cells in a mouse beginning to develop mutations, right, and they're dividing and dividing and dividing, the mouse itself is not going to stick around long enough to worry about that becoming full-blown cancer. What about the other extreme? Here's another mammal, an elephant. Body size of an elephant is about 77-fold larger than a human. So that means, roughly speaking, for argument's sake, 77 times greater the number of cells to worry about in an elephant's body becoming cancer. At the same time, the lifespan of an elephant is somewhat comparable to that of a person. Right? So that means an elephant sticks around long enough, just like a person, where many mutations could be building up in transforming cells, and yet you have way more cells to worry about that could actually be going down this path of becoming cancer. So strikingly then, the elephant's telomeres are not somehow more aggressively short than that of a human's. In fact, an elephant's telomeres are quite similar in length. That means the molecular clock of a fully differentiated cell inside of an elephant undergoing division, right, is about as strenuously controlled as that of a cell in our body. So how then has the elephant evolved to cope with having many more cells in their body, so many more opportunities for mutations, right? And yet their telomere size is about the same, and they live about as long as we do. This has to do then with our discussion of tumor suppressor genes. Remember that one of the most important tumor suppressor genes that we have in our body is P53, the guardian of the genome that can actually halt the cell cycle and trigger apoptosis if things are going wrong inside of a cell. So you'll recall that the human genome has one copy of P53 on chromosome 17. So you can either say you have one copy, or technically speaking, you have two copies, because you have one chromosome 17 from mom and one chromosome 17 from dad. right? So you might say you actually have 2 copies of p53, right? Which is what we just talked about earlier when we were talking about loss of heterozygosity in p53 due to point mutations that by chance can mess up the function of p53 in tumors. An elephant, then, you might be surprised to find out, has 20 copies of the p53 gene in its genome. So in other words, each elephant cell has 40 copies, 20 from its mom, 20 from its dad, right? And that's how the elephant makes sure that there's plenty of p53 in its cells to halt the cells that are going down the wrong path of becoming cancer. And importantly, because there's 40 copies of p53, that means even if some of those p53 genes get a hit in the DNA binding domain through a point mutation, there are enough good wild type versions of the protein still being made by the elephant cell, so you don't have that same problem as much of having when the p53 proteins come together as a tetramer on the DNA, right? The elephant has so many copies of good p53 that even if a couple of them are bad, overall those tetramers are not going to be as messed up compared to in humans, where if you had one bad and one good copy of p53, as we learned, you have problems. Okay, so that really emphasizes then the importance of p53 in fighting cancer, and the fact that it's actually used and relied upon in other mammals as well.